Good morning, dear students. Under the chapter of molecular biology, so far you have studied the two important processes, that is the replication and the transcription. Now, before we go for the translation process, let us study in detail about two important concepts, that is genetic code and mutations. So the content for today's class is going to be genetic code and mutations, which I shall be doing under two sessions. The specific learning objectives for today's class will be, at the end of this session, you should be able to define codon, discuss the organization of genetic code, name the initiator and terminator codons, discuss the characteristics features of genetic code, define mutation, classify mutations, classify point mutations based on the type of nucleotide altered, categorize point mutations based on consequences, citing examples, and explain the frame shift mutations and its consequences in protein biosynthesis. As I said, I shall be doing this in two sessions. In the first session, let us study the genetic code. So it is the first four specific learning objectives concerned with the genetic code I shall be explaining in this particular session. We have studied that the letters A, G, T and C represents the respective nucleotides and found in the DNA. Now, these nucleotides A, G, T and C are present as a three-letter code called codon in the protein coding genes. That is, in the DNA, in the genes, they are organized into three-letter codes called as codons. So, what is a codon? A codon is defined as a trinucleotide sequence which codes for an amino acid. That is, three consecutive bases are arranged to form a codon which codes for a specific amino acids. So, collection of these codons makes up the genetic code. So, what is genetic code? It is a collection of codons. What is a codon? Codon is a trinucleotide sequence which code for an amino acids. So consecutive three nucleotides arranged together forms a codon. The genetic information is stored in the arrangement of these codons. That is, genetic information resides in the order of bases, which in turn are arranged as codons. From here you can see that this is a DNA. A part of the DNA has been transcribed to form the mRNA molecule with the nucleotide sequence which is complementary to this part of the DNA. Now these nucleotides in the mRNA are arranged as a trinucleotide sequence of codons. That is, ATG is one codon, CTA another, GGC, like that codons, and each of this codon will be specifying an amino acids. That is, sequence of the codons specify the amino acid sequence of the encoded proteins. In other words, codons are the key to the translation of genetic information, directing the synthesis of specific protein. There are 4 nucleotides A, T, G and C, and there are 20 standard amino acids. So how do we account for coding of these amino acids by only 4 nucleotides? So the answer lies in the combination of these nucleotides which can code for the amino acids. A combination of 2 can result in 8 codons, which again cannot complete the coding of all the 20 standard amino acids. Whereas a combination of 3, that is a 4 nucleotide in a combination of 3, can give rise to 64 combinations. So 4 to the power of 3 is 64. So therefore, in the genetic code, there are 64 codons. That is, the 4 nucleotides in a combination of 3 results in 64 combinations. Hence, 64 codons are present. Now, of these 64 codons, 61 code for 20 standard amino acids. 3 of them do not code. These are called as terminator codons, also called as stop codons or nonsense codons. They are called as terminator codons, that is a better terminology, because they terminate the protein biosynthesis. So these 3 terminator codons are UAA, UAG and UGA. We also have an initiator codon, that is AUG. AUG, which also codes for methionine. In most of the instances, it also is the initiator codon. What it means is the protein biosynthesis to initiate the first first amino acid which gets added is methionine, which is coded by AUG. In some circumstances, even GUG can also act as initiator codon. Now these codons or the genetic code has directionality. What it means here is that is like the DNA and RNA, even the genetic code is read in a 5' to 3' direction. So this is a table showing the genetic code. So you can see that the 64 codons can be generated by taking the combination like this. That is the first nucleotide can be U, C, A and G, and the second letter can be again U, C, A and G, and again the third nucleotide is U, C, A, G. So a combination of first, second and a third can result in triple U like this one. And this table completely shows all the 64 codons along showing the stop codons over here as well as the initiator codon, and also you can see that the various codons which codes for the amino acids also has been denoted. You need not by heart this table. You need not try to generate this table in the examinations. It's not necessary that you need to write all the 64 codons, so don't waste your time trying to rewrite the table. Coming to the features of genetic code is degenerate, unambiguous, non-overlapping, not punctuated, and universal. Let us study one by one all the different features of the genetic code. So what does degenerate mean? We know that there are 64 codons in the genetic code, out of which 61 will code for 20 amino acids. So that means an amino acid may have more than one codon. So a degeneracy says here is that in the genetic code, multiple codons code for the same amino acid. Now the degeneracy of genetic code is not uniform. What does it mean? It is not like you have 61 codons and you divide it by 20 and each of the amino acid will have the corresponding number of codons. No. What it means here is it is not uniform. It means is there may be certain amino acids which may have more than one codon, up to 6 codons also. For example, leucine, serine and arginine have got 6 codons. There may be some amino acid like glycine which has got 4, and there are certain amino acids which may have 2 or 3. And there are certain amino acids which has got only one codon. For example, tryptophan and methionine has got only one codon. That means the 20 standard amino acids may have the codons anywhere from 1 to 6. So the degeneracy of genetic code occurs because the third nucleotide sequence in the codon is not important in determining the specific amino acid. It is only the first two nucleotides which are important in determining which is the amino acid it is going to code for. So most of the degeneracy of the genetic code is because of the less importance in determining of the amino acid by the third nucleotide sequence. Now the importance of this degeneracy here is degeneracy minimizes the effects of mutations. We will be seeing this how degeneracy can minimize the effects of mutations in the next part of the session. Another important characteristic feature of genetic code is it is unambiguous or specific. What it means is a specific codon codes for only a single amino acid and it does not code for any other amino acid. Now these two concepts, that is unambiguous and degeneracy, should not be confused. Let me explain this with the help of an example. The codon triple U, that is U, U and U, codes for phenylalanine. I'll write it as PHE, which stands for phenylalanine. So this triple U codes for phenylalanine but does not code for any other amino acid. Whereas phenylalanine can also be coded by another codon, that is UU and C. So phenylalanine has got two codons, UUU and UUC. This explains the degeneracy. That is, an amino acid can be coded by more than one codon. Whereas, what does unambiguous say? UUU codes only for phenylalanine, and similarly, UUC codes for only phenylalanine but does not code for any other amino acids. I hope with this, the concept of these two things, degeneracy and unambiguous, is clear to you. So please do not confuse these two important characteristic feature of genetic code. Now, genetic code is non-overlapping. That is, once the reading frame starts for translation process, the codons are read continuously without any overlap of the codons. For example, you have the sequence on the mRNA AUG, which corresponds to initiator codon, and you have rest of the other nucleotides also arranged. So this is read in a continuous manner of triplets like this: AUG, CUA, GGC, triple U, etc. That is, there is no overlap between this. It's not like AUG and then you start with again GCU like that. Okay, so in a sequence of 3, it is continuously read. So that means genetic code is non-overlapping. And genetic code is also not punctuated. So once the reading frame begins, it is read continuously as a trinucleotide sequence. That is, the translating machinery continuously reads it as trinucleotide sequence, and there is no comma or any other punctuation mark denoting that this is one codon and this is the next codon like this. For example, if you have this particular sequence, again CUU, AGC, etc., it is continuously read here. So here for our ease, it has been shown as in a sequence of trinucleotide sequence of codon like this. But in the actual reading frame, there is no comma over here, there is no full stop or any other kind of indication that this is one particular codon and this is the next one is this. There is nothing like that. Okay, so this is what is that genetic code is not punctuated. The genetic code is universal. What it means is a particular codon will code for the same amino acids in all species in the universe. For example, if you take triple U, which codes for phenylalanine, it codes for phenylalanine not only in bacteria but also in humans. So anywhere from bacteria to buffalo, from E. coli to elephant, the same codon codes for the same amino acid. So this shows that probably life originated from a single origin and the genetic code has been preserved. Now, recently exceptions have been noted in the mitochondria. Mitochondria, which houses a small amount of DNA, about 0.1%, has shown a genetic code which may be slightly different from the nuclear genetic code. For example, AUA, which codes for isoleucine in the genetic code of the nucleus, is coding for methionine in the mitochondria. So AUA in mitochondria, it is for methionine, whereas in the nuclear DNA, it is isoleucine. Similarly, UGA, which is a stop codon, codes for tryptophan in the mitochondria, and CUA, which is for leucine, codes for threonine in case of mitochondria. Other two exceptions are AGA and AGG, which are the codons of arginine will code for as stop codon in the mitochondria. So I have just shown the codons, these codons which is coding for the respective amino acids in the mitochondria. You can add here the actual amino acid which is being coded in the nuclear genetic code. Now let us study an important concept called as WOBBLE hypothesis. We studied under the degeneracy of genetic code that an amino acid may have more than one codon. That is, the number of codons vary for an amino acid. It can be anywhere from 1 to 6. And we also saw that it is the first two nucleotide which is important in determining which amino acid a codon is going to specify. Whereas the third nucleotide in the codon is not important in specifying the amino acid. For example, if you take glycine, so glycine has got 4 codons, and you can see that in these 4 codons, the first two is same, GG same for all the 4 codons, whereas the third one can vary, that is it can be U or C or A or it can be G. Analysis of this genetic code has shown that the codons XYU and XYC usually codes for the same amino acids. Okay, so if the third nucleotide sequence is U or C, both may be coding for the same amino acid. And similarly, XYA and XYG will code for same amino acids. Sometimes all four XYU, XYC, XYA and XYG may be coding for the same amino acid, as you can see from the previous example of glycine. Or it could be XYA and XYG coding for one amino acid, and XYU and XYC is coding for another amino acid. So degeneracy of genetic code resides in the third nucleotide of the codon. So this we already studied. Now this codon present in the mRNA will base pair with the anticodon in the tRNA molecules. So this will explain this wobble hypothesis. To understand this, let us see the base pairing between the tRNA and the mRNA, that is the interaction of codon and anticodon. So this is the mRNA which has got a codon, given example here as UCG. Okay, so remember that the genetic code has got directionality, that is it should be always read as from 5' to 3' position. So you have UCG, which the first nucleotide here is U and the last nucleotide is G. And this is the tRNA with its anticodon corresponding to the codon over here. And if we read from 5' to the 3' direction, this becomes the first nucleotide, that is UGA. So from 5' to 3', this has to be read as UGA, whereas this is read as UCG. So what it means here is, that is the first nucleotide of the codon will base pair with the third nucleotide of the anticodon in the tRNA molecule, and third nucleotide of the codon will base pair with the first nucleotide of the anticodon arm. So please understand this, that is first and third, second, and third and first. Now with this background, let us understand the concept of wobble hypothesis. The base pairing between the last nucleotide of the codon and the corresponding nucleotide of the anticodon is not strictly by the Watson and Crick rule. Recall the Watson and Crick rule of base pairing. A base pairs with T, or in case of RNA it is U, and similarly U base pairs with A, and G base pairs with C, and C base pairs with G. Now the interaction between the last nucleotide of the codon and the first nucleotide of the anticodon is not strictly by the Watson-Crick rule. So this is called as wobble. That is, the pairing of the codon and anticodon can wobble at this specific nucleotide to nucleotide pairing site. Let us understand a bit more in detail. So we have this box over here showing the traditional base pairing between the codon and the anticodon. So A base pairs with U, G base pairs with C, U base pairs with A, and C base pairs with G. So this is the Watson and Crick rule of base pairing. Whereas in case of this degeneracy over here is because the third nucleotide of this and the corresponding nucleotide of the anticodon, the base pairing may not be strictly by Watson and Crick. So a nucleotide here at this position can base pair with more than one base of the codon, and it is not strictly by Watson and Crick. So you can see that if at this position, that is at the position of the first nucleotide in the tRNA, if it is A or C, they will always base pair with the corresponding nucleotide only. That is, A base pairs with U only and C base pairs with G. Whereas if the position here is occupied by G, G can base pair not only with C but can also base pair with U. Similarly, if it is occupied by U, U can base pair not only with A but can also base pair with A. Also recall that in the tRNA we have other bases also present. So like for example, hypoxanthine is present, which is denoted as inosine monophosphate with the I over here. So if at this position, that is at the position of this first nucleotide of the anticodon, if it is occupied by this I, that is hypoxanthine nucleotide, now I can base pair in the codon of U also, C also, and A also. That is, it can form base pairings with UCA. So this is the non-Watson Crick type of base pairing. So A can base pair only with U, C can base pair only with G, but G can base pair with C and U also. U can base pair with A and G also. And if it is a hypoxanthine base, the anticodon, it can base pair with U also, C and A also. So that means that a tRNA can recognize more than one codon. So for example, you have this anticodon having UCU, which strictly speaking should form a base pairing with the codon of the mRNA AGA, which codes for arginine. But this UCU can also base pair with AGG, which also codes for arginine. So what it means over here is a single tRNA can recognize two codons, AGA also as well as AGG. Both of them will be coding for the same amino acid. Remember, the first two nucleotide is important for determining the amino acid. So AG is common, but the third position can be G also or A also. Similarly, these three codons, GGU, GGC and GGA, all coding for glycine, can be recognized by the anticodon CCI, I corresponding to hypoxanthine nucleotide. So I can base pair with C also, U also, as well as A. Now, based on these observations, Crick proposed a set of four relationships called as WOBBLE hypothesis. Now, what are these four relationships? Let us see. The first one, what it says is the first two bases of codon always base pairs with the corresponding bases in the anticodon as per Watson and Crick base pairing rule. Whereas the first base of anticodon, which forms a complementary base with the third nucleotide of the codon, determines the number of codons recognized by tRNA. So the first base of anticodon here is determining whether a particular tRNA is going to recognize only one codon or even more than one codon. If at this position, that is the first base of anticodon, has A or C at its side, then it will recognize only one codon. So base pairing here is specific. A will always base pair with U only of the codon, and C will always base pair with G of the codon, and hence the tRNA having A or C at the first base of the anticodon will recognize only one codon. So one tRNA for one codon this. Whereas if at this particular position, if there is G or U, then it can recognize two different codons. So base pairing here between G is not only with C, it is also with U. So G can base pair with C or U, and similarly U can base pair with A or G, and hence a tRNA having G or U at its first nucleotide in the anticodon can recognize two different codons of the mRNA molecule. And if at this position, if there is a inosine, that is a hypoxanthine nucleotide, then inosine can base pair with U, C and A, and hence can recognize three different codons. So it can recognize three different codons. So what it means here is for the 61 codons present in the genetic code, it is not necessary that we should have 61 tRNAs. One tRNA can recognize more than one codon also, depending on the nucleotide present in its first position of the anticodon. Now let us consider the third statement. What it says here is when an amino acid is specified by several different codons, and if the bases differ in the first or second position, then these codons are recognized by different tRNAs. Let us understand this concept with the help of these two codons, AGA and CGA, which differ in the nucleotide in the first position, and both these codons code for the amino acid arginine. Now these two codons require two different tRNA molecules. They cannot be recognized by a single tRNA because the change here is in the first nucleotide. So any change in the first nucleotide or second nucleotide you require a different tRNA molecule. Now the last statement says in a cell, a minimum of 32 tRNAs are required to translate all the 61 codons. So these are the 4 statements which are proposed by Francis Crick, and they're called as the WOBBLE hypothesis. To summarize, codon is a trinucleotide sequence which codes for a specific amino acid. There are 64 codons present in the genetic code, of which 61 codes for the 20 standard amino acids, and 3 of them are called as the stop codons or terminator codons, that is UAA, UAG and UGA. Now genetic code is degenerate, unambiguous, universal, non-overlapping and not punctuated. The degeneracy of genetic code resides in the last nucleotide. And according to WOBBLE hypothesis, the base pairing between the third nucleotide of the codon and the corresponding nucleotide in the anticodon may not be strictly based on Watson and Crick base pairing rule. So with that, we have come to the end of this particular session. So let us take a small break and then we come back and study the mutations in detail.